Mitosis, a process of nuclear division of eukaryotic cells, conventionally divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis conserves chromosome number by allocating replicated chromosomes equally to each of the daughter nuclei. Interphase. Mitotic cell division alternates with a much longer stage called interphase, which often accounts for about 90% of the cell cycle. Interphase can be divided into subphases: the G1 phase, the S phase, and the G2 phase. In G1, the cell grows. In S, the cell continues to grow as it copies its chromosomes. In G2, a nuclear envelope encloses the nucleus. 2 centrosomes are formed by duplication of a single centrosome. Centrosomes are regions in animal cells that organize the microtubules of the spindle. Each centrosome contains 2 centrioles. Chromosomes duplicated during S phase cannot be seen individually because they have not yet condensed. Prophase. The chromatin fibers condensing into discrete chromosomes observable with a light microscope. Nucleoli disappear, and each duplicated chromosome appears as 2 identical sister chromatids joined at their centromeres, and in some species all along their arms by cohesins. The mitotic spindle begins to form. It is composed of the centrosomes and microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosomes are called asters. Prometaphase. The nuclear envelope fragments. The microtubules extending from each centrosome can now invade the nuclear area. The chromosomes have become even more condensed. Each of the 2 chromatids of each chromosome now has a kinetochore, a specialized protein structure at the centromere. Some of the microtubules attach to the kinetochore. Non-kinetochore microtubules interact with those from the opposite pole of the spindle. Metaphase. The centrosomes are now at opposite poles of the cell. The chromosomes convene at metaphase plate, a plane that is equidistant between the spindle's 2 poles. The chromosome centromeres lie at the metaphase plate. For each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules running from opposite poles. Anaphase. Anaphase is the shortest stage of mitosis, often lasting only a few minutes. Anaphase begins when the cohesion proteins are cleaved. This allows the 2 sister chromatids of each pair to part suddenly. Each chromatid thus becomes a full-fledged chromosome. The 2 liberated daughter chromosomes begin moving toward opposite ends of the cell as their kinetochore microtubules shorten. Because these microtubules are attached to the centromere region, the chromosomes have moved centromere first. The cell elongates as the non-kinetochore microtubules lengthen. By the end of anaphase, the 2 ends of the cells have equivalent and complete collections of microsomes. Telophase. 2 daughter nuclei form in the cell. Nuclear envelopes arise from fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. Nucleoli reappear. The chromosomes become less condensed. Any remaining spindle microtubules are depolymerized. Mitosis, the division of one nucleus into 2 genetically identical nuclei, is now complete. Cytokinesis. The division of the cytoplasm is usually well underway by late telophase, so the 2 daughter cells appear shortly after the end of mitosis. In animal cells, cytokinesis involves the formation of a cleavage furrow, which inches the cell into
A modified type of cell division in sexually reproducing organisms consisting of 2 rounds of cell division but only 1 round of DNA replication. It results in cells with half the number of chromosome sets as the interphase. The pair of homologous chromosomes in the diploid parent cell duplicate. This results in a diploid cell complete with the duplicated sets of chromosomes known as sister chromatids. Meiosis 1. The first division of a 2-stage process of cell division in sexually reproducing organisms that results in cells with half the number of chromosome sets as the original cell. Prophase 1. Chromosomes begin to condense and homologs loosely pair along their lengths aligned gene by gene. Paired homologs become physically connected to each other along their lengths by a zipper-like protein structure, the synaptonemal complex. This state is called synapsis. Synapsis ends with the disassembly of the synaptonemal complex in mid-prophase, and the chromosomes in each pair have moved apart slightly. Centrosome movement, spindle formation, and nuclear envelope breakdown occurs, as in mitosis. Metaphase 1. Pairs of homologous chromosomes are now arranged at the metaphase plate, with 1 chromosome in each pair facing each pole. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. Those of the other homolog are attached to microtubules from the opposite. Anaphase 1. Breakdown of proteins responsible for sister chromatid arms allows homologs to separate. The homologs move toward opposite poles guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit toward the same. Beginning of telophase 1, each half of the cell has a completed haploid set of duplicated chromosomes. Each chromosome is composed of 2 sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis usually occurs simultaneously with telophase 1, forming 2 haploid daughter cells. In animal cells like these, a cleavage furrow forms. In some species, chromosomes decondense and nuclear envelopes form. No chromosome duplication occurs between meiosis 1 and meiosis 2. The second division of a two-stage process of cell division in sexually reproducing organisms that results in cells with half the number of chromosome sets as the original cell. Prophase 2. A spindle apparatus forms. In late prophase 2, chromosomes, each still composed of two chromatids associated at the centromere, move toward the metaphase. Metaphase 2. The chromosomes are positioned at the metaphase plate as in mitosis. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from Anaphase 2. Breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move toward opposite poles as individual. Telophase 2 and cytokinesis. Nuclei form. The chromosomes begin decondensing and cytokinesis occurs. The meiotic division of one parent cell produces 4 daughter cells, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distinct from one another and from the parent cell.